Hello once again. In Chapter 4, we're going to discuss the bacteria and archaea. And the article at the beginning of the chapter is talking about plastics that are found in our waste systems. This certainly shows a picture of plastics in the ocean, and this has become a great concern for a lot of people. So, what could be an answer to all of the plastics? Well, this may actually be the answer right here, and that is bacteria that can actually break down the plastics no matter where they're found. So this would be a very important bioremediation if we could find those bacteria that could actually eat the plastics for us. Now, bacteria and archaea versus eukaryotes. Bacteria and archaea both are prokaryotic cells, and one of the big differences between those and eukaryotic cells is how the DNA is packaged within these prokaryotic cells. Bacteria and archaea have nuclear material. They both contain DNA. However, the DNA is free within the cytoplasm. It typically tends to conglomerate towards the center of the cell, and this area is known then as the nucleoid. While eukaryotes have that membrane-bound nucleus that surrounds the DNA of the cell. The cell wall makeup in bacteria, the cell wall is made up of a protein sugar structure known as peptidoglycan, and we'll talk about the shape of that here in this chapter. While the archaea have cell walls that are distinct from bacteria and eukaryotes that have cell walls. They do not contain peptidoglycan. Internal structures, the bacteria and archaea also have no membrane-bound organelles. However, they do have ribosomes that make their proteins. This is just the generalized structure of a bacterial cell. In all bacteria, they all have a cytoplasmic or cell membrane, which is made up of a layer of phospholipids. The bacterial chromosome or nucleoid. In most bacteria, the chromosome is a single circular haploid chromosome. There are a few bacteria that do have linear chromosomes, but they are very few. The ribosomes are scattered throughout the cytoplasm, and this is where we will make our proteins, and of course the cytoplasm itself. In some bacteria, they have an S layer, which is a monolayer of protein used for protection and or attachment in the environment. Some bacteria have fimbriae that are fine hair-like bristles that extend from the cell surface that also help in attachment in the environment. Some gram-negatives will have an outer phospholipid membrane, and this is similar to the cell membrane, but contains a lot of lipopolysaccharides. And we'll talk about the function of it when we talk about the gram-negative bacteria. The cell wall, this is going to be made up of primarily peptidoglycan, and the difference between gram-positives and gram-negatives is the amount of peptidoglycan that's found there. Most bacteria have an active cytoskeleton made up of proteins, similar to eukaryotic cells. Some bacteria can actually form something called a pilus, which is a hollow tube that is used in the exchange of DNA from one bacterium to another. Some will actually produce a capsule that is a very organized layer of proteins and sugars. Some will have inclusion granules, such as droplets of fat or phosphate or glycogen deposits. Some will actually develop microcompartments that are protein-coated packets that are used to localize enzymes and other proteins. Some bacteria will have plasmids. Plasmids are double-stranded DNA circles that contain at most a few genes. These are not considered necessary for life of the bacterium, but may actually impart certain characteristics that are important for survival. And then some bacteria may have one or more flagella, and these can be used in movement and in attachment in the environment. Some bacteria can actually form endospores, and we'll talk about those, and some can actually form intracellular membranes. So be familiar with the structure of the bacterial cell. Bacteria are always single-celled organisms, and they are capable of carrying out all the necessary life functions of a living organism. They reproduce, they do metabolism, they can process nutrients. Bacteria, though, can also act as a group within colonies and in biofilms. One of the things that we look at in classifying bacteria is their arrangement, size, shape, and how everything fits together. The average size of most bacteria is around 1 micron in diameter. There are some, however, larger bacteria, such as this Theomargarita nambiensis, which can range almost up to 800 micrometers. And then the smallest of the bacteria are actually the only wall-less bacteria as well, and these are the mycoplasms. And they actually average about 0.15 to about 0.3 microns in diameter. So this one might be almost 0.5 micrometers, but then these other ones are certainly smaller. And then there are a few of what we call nanobacteria that are even smaller than the mycoplasms that are fairly newly discovered. There are three general shapes of bacterial cells. They can be coccus, which are typically spherical in shape, or they can be slightly oval or bean-shaped and may even have a pointed end. Bacillus, which are cylindrical rod-shaped bacteria. And then the spiral organisms, of which there are three subcategories. Vibrios have a slight curvature along the long axis, and so they typically come off as being quite comma-shaped. Spirillums are thick and rigid spirals where they only have about 3 to 5 twists along the long axis. And then the spirochete are very thin and very highly flexible and are highly twisted. So when we talk about the spiral organisms, the question is, how twisted actually are you? And here's a picture. So again, the Vibrio have about a half turn along the long axis, so they form this kind of comma shape. The Spirillum have, again, about 3 to 5 twists, and then these spirochetes typically have are very highly twisted along the long axis. And I'll explain why they actually are twisted a little later in the lecture. Now, some bacteria are what we call plemorphic, and pleo means many, morpho body. And so we see a variation in size and shapes among the cells within a single species. We have some bacteria known as Coccobacillus. Sometimes they look more cocci-like, sometimes they look more like bacilli, or sometimes they just kind of have a weird shape. And so we refer to these organisms as plemorphic. So here's some of the cocci. So again, the vast majority are going to be just a spherical shape, but some may have kind of a teardrop shape to them, or maybe be a little oval shaped, but not elongated is the main message. The bacilli, they're basically longer than they are wide, and typically they're rod-shaped. But again, they may take on kind of a almost UFO type looking shape, or they might have little clubs at the end. Typically, if I'm going to see all of these shapes in an area of a single organism, then I would call it plemorphic. The vibrio of the spiral organisms, hopefully you can kind of see they've kind of got a little comma shape to them. The spirillums, fairly elongated, and again about 3 to 5 twists along the long axis. And then those spirochetes that are highly twisted organisms. There are a small group of bacteria known as the branching bacteria, and these actually grow in filamentous structures that look very, very similar actually to the eukaryotic fungi, but they actually are truly bacteria. We really won't be discussing these as most of them are not going to be pathogenic, but I just wanted to point them out. Arrangements of the bacteria is another way that we can classify the organisms. So in the cocci, we can have Diplococcus, which is a pair, di meaning two. Staphylococcus, and staphylo actually means cluster or grape-like cluster. And then the streptococci. These are chains. Literally looks like pearls on a necklace. So this can be a chain of few to even sometimes hundreds of cells. The cocci could also actually fit into a box-like structure called the tetrad, which is a layer of 4 cocci stuck together, or the sarcinae, which is a cubital packet of 8, 16, or more cocci. And really the sarcinae is simply 2 tetrads or more set on top of each other, forming a true box. So here's a good picture of some Diplococcus. Typically, these are cells that have undergone cellular reproduction and actually just stick together. The Streptococcus are chains. Here's a good example of a tetrad, 1, 2, 3, 4 in a layer. The Sarcinae, a little harder to tell on this particular one, but again, usually they form kind of a cube-like structure with 8. And then the Staphylococcus are these grape-like structures. In the bacilli, we see diplobacilli, which are pairs of the 2 rods stuck together, and streptobacillus, which is chains of the cells. We do not see the tetrads or the staphylo arrangement in the bacillus. Sometimes, however, the bacillus can form palisades, which are cells of a chain that remain partially attached and simply kind of fold back and forth, creating a side-by-side row of cells. The sporilium occasionally will form short chains, but it's fairly unusual, and the spirochetes rarely remain attached after cell division. So typically the spiral organisms, including the Vibrio, will all be single cells. So here we have our arrangements of our bacilli. And here's our diplobacilli and the streptobacilli. Anytime you see more than 3 bacilli together, we refer to it as strepto versus diplobacillus. And then this actually shows something known as a coccobacillus. Again, they tend to be kind of short rods, and oftentimes they form diplopairs, but sometimes they actually are more spherical in shape, even though this is all the same organisms. So a lot of times I would actually even say coccobacillus are just simply pleomorphic bacteria. So here's our first concept check. You should be able to describe at least 3 structures common to all bacteria. You should be able to describe at least 4 structures found in all bacteria and at least some of those that are found in some of the bacteria. You should be able to know the 3 general shapes of bacteria and to describe the arrangements found among those general shapes of bacteria. So let's start with the external bacterial structures. As I said, some bacteria may have what's known as this S layer. And this is just simply thousands of copies of a single protein that are linked together. It provides protection from environmental conditions such as drying out, and it typically is only produced in hostile environments. When pH changes, it gets too hot, it gets too cold. So really, we're not going to discuss a lot about the S layer. The glycocalyx, however, is found in most bacteria in one form or the other. They can either produce what's known as the slime layer, which will form a loose protein carbohydrate structure around the outside of the cell, and typically this is fairly thin, and it will protect the cell from loss of water and nutrients primarily. Or they produce a capsule, which is a much more organized, more thickly bound glycocalyx than the slime layer. It's denser and thicker, and therefore a little bit more protective overall. So here's what we see with the slime layer, and again, it's kind of just loosely arranged. Typically under the light microscope, you'll not be able to even see this slime layer because it's fairly thin versus the capsule, which is very thick and compact and will actually usually be seen in a capsular stain. Specialized functions of the glycocalyx. The capsules actually are very important for those cells that can form them, because they are formed by most pathogenic bacteria, and it protects the bacteria against the phagocytic white blood cells of the immune response. Basically, what this capsule does, it's kind of like an invisibility cloak for the immune system, so that the immune system can't see this particular bacterium, so therefore it is hidden from immune response. Now, biofilms actually are communities of organisms that work together for the good of all. They're kind of like a commune back from the 60s. And a good example of this would be plaque on teeth surfaces. So there can be actually many different organisms, not just bacteria, but even other organisms as well, that may be producing one may produce something that another one needs in the group, or certainly just the large clumping of the cells together can provide protection, especially for the inner cells. And so these biofilms actually do a lot for each individual of the biofilm. Problem is that a lot of times these biofilms will form on hard surfaces, and that will actually include long-term indwelling artificial devices that are implanted. And this can be a real problem with the failure of some of these devices, such as artificial joints. Two major groups of appendages that we can find on bacteria: the flagella and other axial filaments that are similar. And again, here's some pictures of these flagella in a flagellar stain. These can provide motility as well as adhesions in the environment. The fimbriae, the pila, and the nanowires. I'll talk about the fimbria first. These actually will provide attachment points within the environment, either to other bacterial cells or to cells within the environment, and the pili will be for the exchange of genetic material. Now this picture actually shows these little nanowires, and these are somewhat probably similar in structure to fimbriae, but they're very, very short, and they actually can stick this cell down into its environment and may actually even form channels between cells. The prokaryotic flagella is different from the eukaryotic flagella in several different ways, and I'll explain this in just a moment. These are located outside the cell wall. They are anchored actually into the cell wall and the plasma membrane by what's known as the basal body. The filament itself is made up of chains of a protein called flagellin. It is attached to the basal body by the hook, which actually will provide the rotation of the flagella filament. Prokaryotic flagella rotate, similar to the propeller on a motorboat. So they actually are moving in a rotational movement. This is different than eukaryotic flagella, which actually undulate back and forth in this manner. So we'll talk about the eukaryotic flagella when we get to the next chapter. This is a little better demonstration of what the actual flagella looks like. So here's our basal body, and depending upon whether you're in a gram-positive or gram-negative bacteria, you'll have either 2 or 4 of these rings within the basal body. They'll be surrounded by another type of protein that helps anchor into the plasma membrane and the cell wall itself. This actually is a rotor motor, and I like this electron micrograph of the actual basal body here. Then we have the hook that again is going to provide the propeller-like motion of the filament, and then the filament itself. Now, how the flagella are arranged around the bacterium is also a way that we can classify the bacteria. Polar flagella simply means it's attached at one or both ends of the elongated cell. And so we see monotrichosis. This would be a single flagellum located at one end of the cell. Lophotrichosis means that we have more than one flagella, usually in bunches or tufts, located again at one pole or one end of the cell. And then amphitrichosis, amphi meaning both ends, basically, we have flagella at both poles of the cell. The fourth arrangement of flagella around bacteria is what we call peritrichosis. And this is where the flagella are dispersed randomly over the surface of the cell. So here's what these look like. So peritrichosis, here's our cell, and these flagella are just kind of located all the way around the cell. Monotrichosis, it's on one end, it's single, so the mono. So we usually say they're monotrichosis and polar, even though we really don't need to say that because we know it's on one end and that's it. Lophotrichosis, again, located on one pole of the cell, but now we have more than one flagella. And then amphitrichosis, and these are often found on the spirillum and spirochetes, and these are actually maybe 1 to 2 that are found on both ends. Now, while the monotrichosis, lophotrichosis, and peritrichosis are oftentimes used in cell movement, the amphitrichosis are actually more like rudders on the boat. The spiral organisms are going to move in a spiral fashion, and these are simply there for guidance through the environment. What's chemotaxis? Well, chemotaxis is movement in response to chemical signals within the environment. And we can have positive chemotaxis, where the movement of the cell is in the direction of the chemical stimulus, or negative chemotaxis, where basically the chemical will be a repellent and the cells will move away from whatever the chemical is. Phototaxis means that we're moving towards light. And so a lot of our photosynthetic bacteria will actually move towards light so that they can utilize it to make their energy. So how do the bacteria use the flagella to move? Well, in the monotrichosis and lophotrichosis, we can see since it's all on one end, it's like having all of your motors on the end of your boat and you're going to go forward. But what about the peritrichosis? This is like you've got motors all the way around your boat and they're all pointed in different directions. Well, sometimes the flagella kind of get organized and they all tend to be pointed in one direction. And this is when the bacteria will do what we call a run. This is a counterclockwise movement of the flagella, and this actually will then allow the forward momentum of the bacteria towards or away from a stimulus. But at some point then, the flagella reverse direction and basically become uncoordinated, and this causes the cell to stop and actually change course. And so they just kind of tumble through the environment. And oftentimes chemical repellents cause numerous tumbles. Think of a child trying to run away from something they don't want or is dangerous, and oftentimes they're kind of falling down and as because they're hurrying so quickly. Well, that's what sometimes will happen with tumble. So here's what it looks like. So when the flagella are all working together, they kind of get into this one direction and we get a nice little run. And then they reverse direction and we kind of tumble around and then we reverse again and we get a run and then we tumble and then we run. And so it kind of just goes through the environment somewhat erratically. And this actually shows if there's no attractant or repellent, we just get kind of this random movement through the environment. But if there is an attractant, the walk becomes biased towards more runs and fewer tumbles, and therefore we kind of get this forward momentum, even though we're kind of going at it at a stumbling rate with the tumble. Now, we also have to talk about the endoflagellum, and these are found primarily in the spiral organisms. This is known also as the axial filament. This is actually an internal flagellum. Uh, Typically, there'll be 2 or more long coiled threads found just underneath the cell membrane within the cytoplasm of the cell. These actually will be anchored at one end and will have rotational movement throughout the long axis of the cell. What will actually happen is then this imparts a twisting or flexing motion, which does two things. One, gives the characteristic shape of the spirulium or spirochete, and the other thing gives rotational movement of the organism. So when we have these endoflagellum here within the cell, then this will allow again for that twisting motion. And this is a great picture of one of these axial filaments. And so you can see this is what provides that twisting arrangement, as well as then the twisting movement of these organisms in their environment. Now, attachments can enhance pathogenicity in some bacteria, and one of these is the pilus or the pili. These are sometimes referred to as the sex pilus as they are involved in the exchange of genetic material. Now, let me just say this. Bacteria all reproduce asexually, okay? But sometimes we do exchange genetic material. The fimbria or fimbriae is the plural, again, primarily used for attaching the organisms into their environment. And so both provide adhesions but not locomotion. Flagella also can sometimes be used for attachment in some species. In fact, some bacteria have non-modal flagella, meaning they're not used for movement, but they're used for attachment only. The fimbriae, these small bristle-like fibers made of proteins that sprout off the surface of the bacteria in some bacteria. They're kind of short, much more shorter than the flagella. Their composition will vary, but most actually contain just protein structures. And they have the inherent tendency to stick to each other and to other surfaces. They're probably one of the main things responsible for the formation of the biofilms, especially in what we call the monolayer, the first layer of cells that adheres to the surface that begins to form the biofilm. E. coli and the Gynococcus bacteria use fimbriae to actually adhere to epithelial cells in their environment. So here's a good picture of these kind of short little stubby fimbriae. If one of these had a flagellum, it would be like much longer than the fimbriae. And this is actually E. coli, and this is the microvilli of the intestinal mucosal cell, and these fimbriae are actually helping stick those E. coli onto the surface of that epithelial cell. The pilus, again, sometimes known as the sex pilus, even though, yes, again, not bacterial sex. It's simply exchange of genetic material. It's a long, rigid, hollow tube that's made of the protein called pilin. These are found only in gram-negative bacteria, so gram-positives do not produce sex pilus. The pili are used in the process known as conjugation that we'll talk about later. That's where we get the partial transfer of DNA from one cell to another. The production of the pilus is genetically controlled in those cells that can produce it. And so only cells that have something called the F or fertility plasmid can actually produce the sex pilus. Now, they can pass that plasmid onto another cell, and then that cell will become a genetically controlled cell that can produce a pilus as well. So this cell actually produced the sex pilus, and you can see it's just a long hollow tube. And actually what will usually happen is once it attaches to another cell over here, it will actually draw that cell closer, and this will widen the channel within the pilus for the exchange of the genetic material. Typically, the genetic material is a one-way exchange, so it will go from the donor cell to the recipient cell. And once the genetic exchange is done, the pilus will disconnect and from both cells and then just simply float away. Okay, so can you name the three parts of the bacterial flagellum, and can you tell me how it actually moves? What is the difference between a run and a tumble in those peritrichosis bacteria? And true or false, bacterial fimbriae are used in conjugation. And can you describe the differences between the slime layer, the capsule, and biofilms? All right, so now that we've talked about the external structures of the bacteria, let's talk about the actual wall of the bacteria. So something that we sometimes refer to as the cell envelope that is composed of 2 or 3 basic layers. One, the cell wall, the cell membrane, and then the outer membrane that's found in gram-negative bacteria. These act as a single protective unit. Now, there's a difference between the cell wall in the gram-negative and gram-positive cells. And so this is one of the reasons that we use the gram stain in order to identify many of the bacteria. The gram stain was developed in 1884 by Hans Christian Gram, and it delineates the 2 major groups of bacteria, gram-positive, gram-negative. And as I said, in the gram staining, gram-positive stain purple, gram-negatives will stain a reddish color. The difference lies in the structure of the cell envelope, and I simply will say the cell wall. So let's look at the differences. Gram-positives have a thick multi-layer wall composed of many, many layers of this protein called peptidoglycan. It will have the inner cytoplasmic membrane or the cell membrane in the cell envelope. But the main difference is we have a very, very thick wall of peptidoglycan. Whereas in gram-negative bacteria, we find an outer phospholipid membrane that has many, many proteins in it as well as some sugar structures. Then we have a thin layer of peptidoglycan, usually 1 to maybe a few layers of that protein sugar structure. And then finally, again, the cell wall itself, or I'm sorry, the cell membrane itself. So just a pictorial. This is actually layer upon layer upon layer upon layer upon layer of peptidoglycan. This is the bilayer phospholipids in the cell membrane. And how we actually hold all of these layers of peptidoglycan together in the gram-positive cell is through a bunch of what we call teichoic and lipoteichoic acids. It's kind of like glue that holds the layers of a wood laminate together. Whereas in the gram-negative, our peptidoglycan layer is very thin. So that's the pink layer here. Here's our cell membrane. But then we have this outer phospholipid membrane that typically has, again, a lot more proteins. It will have some transport channels in it, lipopolysaccharides. So while it's still a phospholipid membrane similar to the cell membrane, it's different in its other compositions that we see. And the difference between the layers of peptidoglycan is what will make these gram-positive and gram-negative in the gram staining process. So, what is the functions of the cell wall? Well, the biggest function of the cell wall is to keep the cell from exploding. Most bacteria have a very high internal osmotic pressure, and therefore this cell wall kind of acts like a brick wall that will keep the cell from bursting due to that high osmotic pressure. But it will also help determine the shape of the bacterium. So what is this peptidoglycan? Well, it's found in all bacteria, almost all, except for the mycoplasmas. And it's a unique macromolecule composed of glycan chains, which are sugar chains, cross-linked with short peptide fragments. And I'll show you the picture in just a second. This provides a very strong but flexible support framework in the cell wall. So here's what we have. We have 2 sugars, N-acetylglucosamine called NAG an N-acetylmuramic acid called NAM, and they alternate. So we have NAM-NAG, 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 and these form these long rod-shaped structures. And we lay down these rods next to each other, and we actually attach them in together with these short little strands of short little strands of peptides. And so these are simply amino acid chains. This basically then forms a monolayer. In the gram-positives, what we're going to do is then take this monolayer and add another on top and another and another and another and glue them together with the lipoteichoic and teichoic acids to make a thick, strong layer of peptidoglycan. So how thick is the gram-positive cell wall? Well, it's about 20 to 80 nanometers thick. And you think, wow, nanometers, that's not real thick. Well, it is relatively to the gram-negatives. The functions of the teichoic and lipoteichoic acids will be to help maintain the cell wall. Again, it's kind of like the glue that fits all of the wood layers together. It actually will allow enlargement of the cell during cell division. And the teichoic and lipoteichoic acids actually will form an acidic charge on the cell surface. So again, borrowing out of another text, here's our NAG-NAM rods held together by the short little peptides. And then we layer this one and this one and this one and keep going and keep going. And these teichoic and lipoteichoic acids then help hold those multi-layers together. The gram-negative cell wall is only about 1 to 3 nanometers thick. See, told ya. Okay. Single, usually thin sheet, usually single, may have 2 to 3 layers of the peptidoglycan, but certainly not multiple layers of it all. Somewhat rigid in structure. The thinness gives the gram-negative bacteria greater flexibility. However, they are more sensitive, therefore, to lysis due to the high internal osmotic pressure. The gram-negative outer membrane, which is really part of the cell wall, is again a bilayer of phospholipids that contains specialized polysaccharides and proteins. These lipopolysaccharides will function as antigens and receptors on the surface of the cell, and they may actually become what we call endotoxins. And this is one reason gram-negatives can cause a lot of problems, because these endotoxins can actually stimulate fever and shock reactions within their hosts. The lipoproteins will help anchor the outer membrane to the thin layer of peptidoglycan. We also will see that there are a number of poron proteins within the outer membrane of gram-negatives, and these are simply transport proteins that will span the outer membrane, but it only allows relatively small molecules to penetrate. Typically, these are not very specific, and so as long as you're small enough, you can get through these. So size can be altered to block the entrance, however, of harmful chemicals if we come across that in the environment. The porons can also act as a defense against certain antibiotics, because most antibiotics are very large, and so if the antibiotic is going to work inside the cell, they have a very hard time getting into the gram-negative cells. So again, thin layer, single layer of peptidoglycan. We have the cell membrane here, and then this is that outer phospholipid membrane of the gram-negative. And you can see there's a lot more proteins and a lot of these lipid polysaccharides that are floating around in this outer membrane. Now, non-typical cell walls, these lack the cell wall structure of gram-positives or gram-negatives. And the bulk of these cell walls are of the Mycobacterium and Nocardia genuses. Both of these are our acid-fast bacteria. Now, while Mycobacterium and Nocardia cell walls are more similar to gram-positives, they may not be as thick in the amount of peptidoglycan that they have. But what they do possess is something else, and that is called mycolic acid or cord factor. This is a very long chain fatty acid, and it contributes to the overall pathogenicity of both of these genuses of microorganisms. It makes them very resistant to certain chemicals and dyes. And I will say, Mycobacterium and Nocardia will stain a gram positive. It will be a weak gram positive. So to identify these, we actually have to do that acid-fast staining process. And so if I'm suspecting Mycobacterium tuberculosis or Mycobacterium leprae in my patient or one of the Nocardia species, I'm going to ask for an acid-fast stain. The mycoplasmas naturally lack a cell wall. Part of the reason for this is they are very, very small. They are of the smallest bacteria that we find. Also, their cell membrane has a lot more sterol, such as cholesterol, and therefore is resistant to lysis of the cell. Mycoplasmas therefore tend to be pleomorphic in shape, ranging anywhere from about 0.1 to 0.5 microns in diameter, ranging from filamentous in shape to coccus, or even sometimes donut shaped. And so a lot of times, especially when we grow mycoplasmas on artificial media, they actually form these spindle-like cells. And so this is again because of the lack of that cell wall, they actually have more flexibility and will grow in some unique patterns. The cell membrane itself is very similar to the eukaryotic cell membrane. About a 5 to 10 nanometer sheet molded completely around the cytoplasm, made up of the bilayer of phospholipids that have then proteins embedded. About 30 to 40% of the entire cell membrane is made up of the bilayer phospholipids, and another 60 to 70% are actually proteins that can act as primarily transport channels or stability proteins. Mycoplasmas contain the high amount of the sterols, such as cholesterol, and that adds to then the stability of the cell membrane. The archaea contain unique branched hydrocarbons in their cell membranes rather than the fatty acid chains. And so this is one reason why we separated the bacteria from the archaea. Functions of the cell membrane in bacteria, and this is a big one. This is where we're going to see the majority of ATP production, and we'll talk about that when we talk about cellular respiration later on. We'll then see nutrient processing. We have to get the nutrients into the cell in order to produce that energy. And we'll see synthesis of various products at the cell membrane besides ATP. And it is a selectively permeable membrane, so it does regulate transport of things into and out of the cell. So such as a nutrient passing into the cell or discharge of waste products. Because of the selective permeability, water and small uncharged molecules diffuse freely into the cell. But special carrier molecules then exist as passages for other molecules that cannot get through the phospholipid membrane itself. Secretion, typically discharge of metabolic products into the extracellular environment, is also going to be a function of the cell membrane, and there are several different ways this can occur. The outer membrane in the gram-negative bacteria makes them pretty much impervious to antimicrobial chemicals. It is much more difficult to kill gram-negative bacteria using antibiotics than gram-positives. And the reason is this outer phospholipid membrane in the cell wall. Infections with gram-positive bacteria are typically treated differently than the infections with gram-negatives. This is one of the reasons why it's so important that I know, do I have a gram-positive or a gram-negative infection in my patient? It's going to assist me in choosing the therapy that I want to use. The cell envelope is really what interacts with the host patients and cause disease within humans and animals. Proteins in the outer cell wall of gram-positive bacteria can actually become toxic. Lipids in the cell wall of mycobacterium can be harmful to human cells, and primarily this is the mycolic acid. Macromolecules in the cell wall are seen as foreign and can stimulate antibody production by the host's immune system. So, our concept check. Can you describe 3 differences between gram-positives and gram-negatives? Can you describe how the outer membrane of gram-negatives contribute to disease symptoms? And are you able to describe 3 functions of the cell membrane? Okay, so let's go inside the cell. Cytoplasm is cytoplasm. It's a gelatinous solution contained by the cell membrane. It's the prominent site for the cell's biochemical and enzymatic activities. It's approximately 75% or so water, and the rest is going to be a complex mixture of sugar, amino acids, and salts. Also will contain the chromatin, which is making up the DNA, the ribosomes, which are going to make our proteins, and any granules and the fibers that act as the cytoskeletons and any inclusions as well. Now let's look at the bacterial chromosome. As I said, typically most bacteria contain a single circular strand of DNA that is haploid in structure, meaning there's only one copy of each gene. It's aggregated in a dense area called the nucleoid region because we don't have the nuclear membrane. And the DNA is tightly coiled around some basic protein molecules to fit inside the cell component or cell compartment. The DNA is typically much longer overall than the cell itself, so basically we have to wad it up to get it into the cell. Plasmids are non-essential pieces of DNA. Most of the time they contain just a few genes, but these are not essential to the survival of the cell. However, they may confer protective traits to that particular cell. They're important in genetic engineering because we can cut and splice plasmids and move plasmids fairly easily from one cell to another. And so these are actually going to be very important when we talk about genetic engineering. Now, these are typically not reproduced during cellular reproduction. So when the cell separates in reproduction, one cell gets the plasmid and the other cell typically does not. So here's our nucleoid region. So here we've just wadded up this single circular chromosome into kind of the center of the cell. And I love this picture. This is actually E. coli, and this has been treated so that the DNA is being extruded from the cell. So if I started right here and followed this all the way around, I would wind up right back here. But you can see how much more of the DNA there is on trying to get into this little itty bitty living space of the cell. And then this is actually showing in comparison a small plasmid. And so even though this is at a higher magnification, you can see how much smaller they are in comparison to the chromosome itself. The ribosome. These are little protein factories made up of RNA and proteins. They're dispersed throughout the cytoplasm in the prokaryotic cell, and they sometimes can even be found in chains. They are actually made up of 2 subunits, a large subunit and a smaller subunit, and these are actually measured in size called the Svedberg units or the S units. This is a measurement of the relative size of the cell parts through a process known as sedimentation during centrifugation. I won't go into that, but basically the S unit is how much each of these subunits weigh. In the prokaryotic cell, the large subunit is a 50S subunit and the small subunit is a 30S subunit. When we get ready to make the protein, the small and large subunits come together to form the ribosome, and this in prokaryotic cells is called a 70S ribosome. Yes, I know, 50 and 30 do not add up to 70, but think of it this way. When the large and small subunit come together, they slam together and they lose 10 pounds. Oh, I only wish that would happen with me. But this is a 70S ribosome found in the prokaryotic cells. In eukaryotic cells, however, the large subunit is a 60S subunit, the small one is a 40S, and they come together to form the 80S ribosome. And we'll talk a little bit more about them in the next chapter. Inclusion bodies, these are usually storage sites for nutrients during periods of abundance, and this could just simply be a fat droplet that's floating around in the cytoplasm. So they vary in size, number, content, okay. And so these are just droplets of some sort of nutrient in the cell cytoplasm. These are actually small, and these are actually known as magnetomes. These actually contain some iron structures within this bacterial cell. The cytoskeleton are long polymers of proteins that are similar to the eukaryotic protein called actin, so a very thin little protein. They can be arranged in kind of a just a haphazard manner in the cocci and bacillus, but in helical structures, the spiral organisms, they're arranged in a helical formation, and that again gives that kind of common shape. So we see that spiraling of these organisms. So this contributes to the cell shape. Endospores are going to be formed by only certain bacteria, and they are able to withstand hostile conditions and facilitate survival of the organism when the environment is not friendly to the living cell. Let me make this perfectly clear. These are not reproductive spores. These are formed only by those cells that can form them when the environment becomes hostile to the living cell so that the DNA can survive. Two phases that occur in these endospore formers. The vegetative cell is the living, metabolically active cell. These are the ones that are going to be in the environment when it's good. But if the environment changes in some way that is hostile to the vegetative cell, these cells actually will then form the endospore, which is an inert, encapsulated structure that contains the DNA of this organism. Sporulation is when the spores form that is induced by environmental conditions. So here's what happens. Basically, if the environment becomes hostile or we're seeing something changing, these spore formers will actually make a copy of the DNA and then begin to form a protein coat around the copy of the DNA. So the plasma membrane starts to surround the DNA. The spore septum then surrounds the portion, and basically this becomes a double membrane structure. We then lay down peptidoglycan around it, and we form the spore coat. At this point then, typically the vegetative cell is in its death throes, and so the bacterial chromosome is all broken up, and this cell simply dies and will release the endospore. So here's our nice little endospore that's being formed inside the cell. This vegetative cell is going to actually die. These endospores can resist extreme temperatures of heating. It certainly can survive desiccation or drying out. It can survive freezing temperatures. It can survive certain amounts of radiation. It can survive lots and lots and lots of different chemicals. So these endospores are survival mechanisms, not reproductive spores. So why do we autoclave at 121 degrees Celsius for 15 minutes at 15 pounds per square inch? Because we know at that temperature and that pressure, we will actually destroy endospores of any of the spore formers. So this will ensure that all of the endospores are destroyed on things like surgical instruments. Now, when the environment becomes favorable again, okay, so we get exposure to water, and usually there's some sort of germination agent. There's something in the environment that becomes favorable to the living cell again. Germination agent stimulates the formation of hydrolytic enzymes that break down the spore capsule, and the core rehydrates and takes up nutrients, and a single bacterium grows out of the endospore coat. Once initiated, this germination will actually be completed in about 1.5 hours. So we've formed our spore, and what will happen then is, as the water is engulfing or coming back into the spore, it will actually begin to germinate then a new cell. So again, not reproductive, because one cell made the endospore. One cell will be produced from each endospore if the environment becomes favorable again. So what's the medical significance of these endospores? Well, they're constantly intruders for sterility and cleanliness, because they can survive such extreme environments. It's very difficult to completely get rid of all the endospores. That's why we use the autoclave or incineration. So most of these endospores are going to resist any ordinary cleaning methods, such as boiling, soaps, disinfectants, even your hand sanitizers and all of that. They just kind of look at it and go, yeah, right, whatever. Okay. So we get frequent contamination of cultures and media or tissues and surgical suites, etc., by these endospores. Hospitals have to protect the patients against these endospores in wounds, because the wounds provide the perfect environment for many of these endospore formers to actually become vegetative cells and cause disease. So destruction of these endospores is not only important in medical facilities, but it's also important in food production. The reason that tin canned food is heated to such a high temperature is to destroy any endospores that may have been in the cans when they were canned. Some diseases are related to the persistence and resistance of their spores, and there's two primary groups of endospore formers that are pathogenic. That is the Bacillus species, and one of the biggest one is Bacillus anthracis that causes anthrax. And then the Clostridium species, Clostridium tetani that causes tetanus, Clostridium difficile that causes pseudomembranous colitis, also known as C. diff, Clostridium perfringens that causes gas gangrene, and Clostridium botulinum. And in fact, when I mentioned the canned foods, the reason that we actually heat canned foods to such a large or to such a high temperature is to get rid of any endospores of Clostridium botulinum. And this is actually known as commercial sterilization. So, true or false, plasmids are vital to the metabolism of bacterial cells. How do bacterial ribosomes differ from eukaryotic ribosomes? Can you list and describe the steps of endospore formation and germination? And what are 3 diseases caused by endospore forming bacteria? And what are the 2 main genera of bacteria that are endospore formers that cause disease in humans and animals? Okay, let's turn our attention to the archaea. The archaea actually are more closely related to the domain eukarya than actually to the domain bacteria, even though they are prokaryotic cells. They share ribosomal RNA sequences with the domain eukarya that are not found in bacteria. Protein synthesis and the ribosomal subunit structures are more similar between the archaea and eukarya. And this just simply compares the 3 cellular domains. So while both bacteria and archaea are prokaryotic, Okay, and they typically have single or circular chromosomes. Okay, and even though they both have 70S ribosomes, the archaea 70S ribosomes have some structural similarity to the 80S ribosomes found in the eukarya. Okay. Protein synthesis is more similar in the archaea than in the bacteria. Bacteria have peptidoglycan in their cell wall, while the archaea have other proteins, and those eukarya that have cell walls have other either proteins or sugars in their cell wall. And so there's some major differences between the three domains. Differences from other cell types with the archaea is that certain genetic sequences are found only in their ribosomal RNA. And this is one of the main reasons that Woos and Fox actually then uh, supported the formation of the domain system, was because they saw the difference in the ribosomes from bacteria to the archaea. They have a unique method of DNA compaction, and they have unique and chemically distinct cell walls. Primarily, the cell walls of the archaea are made up of something called oglycans instead of peptidoglycans. The archaea, however, are probably the most primitive form of all living life forms on Earth. They're more closely related to the cells that originated some 4 billion years ago on the planet, and they live in habitats that are probably more similar to the extremes that were found in the planet at that time. So very hot, very cold, very salty, very acidic, high pressure, all of those different things. Low oxygen in the atmosphere. In the archaea, there are actually methane producers. There are hyperthermophiles that like it really, really hot. Extreme halophiles that like it really, really salty. Sulfur reducers, and something that we call extreme barophiles that like extreme high pressure. The methanogens actually convert CO2 and hydrogen gas into methane gas, which is CH4. They're common inhabitants in anaerobic swamp mud, the bottom sediments of lake and oceans, and even the digestive systems of some animals. The gas produced in swamps may be a source of fuel, and in fact, even our landfill, in Springfield's landfill, they are actually using methanogens to produce methane gas in the large garbage piles, and they're collecting that to actually use as fuel. However, some of this may contribute overall to the increase in greenhouse gases within the atmosphere. The extreme halophiles require huge amounts of salt in their environment to actually grow and reproduce. They can multiply in environments that have 30% sodium chloride that would actually destroy most other cells. They primarily exist in inland seas, salt lakes, salt mines, and even in salted fish. And this is just a picture of some of these. They can use a red pigment to synthesize ATP in the presence of light, and this is one of the problems with some of these archaeas, is the fact that they can cause these red tides that actually then can cause death to any other living organisms within this area. The psychrophiles are adapted to grow at very low temperatures, and I will say there are some bacteria that are also psychrophiles, and we'll talk about those later on. The hyperthermophiles like to flourish at temperatures between 80 to 113 degrees Celsius. Some of them can even grow as high as 131 degrees Celsius, and they cannot grow below 50 degrees Celsius. I don't worry about these guys because there's no way in the world they're going to exist in our bodies because we're just too cold for them. They typically live in volcanic waters and in soils and submarine vents. They're often also salt and acid tolerant as well as pressure tolerant as well. So many of the archaea are unculturable due to the extreme environments in which they live. If we try to bring these archaea in from their environment into one of our environments, they simply can't live because the conditions are not correct. So the ribosomal RNA sequencing is the most often used in identifying these archaea. And this technique has also advanced the general understanding of protein synthesis and cellular evolution as we've looked. So here we have this concept check. So how are the archaeal cell walls different from the bacterial cell walls? Can you define the terms methanogen, halophile, psychrophile, and hyperthermophile? And true or false, archaea do not cause human disease. Okay, so classifying bacteria and archaea. The classification system serves both practical and academic purposes. One, we can differentiate and identify unknown species, and that's certainly true in the archaea because there's just some that we're just finding out about. We can study the relationship and origin of microbes. So what are some of the characteristics used to trace the origins and evolutionary relationships between these two cells? Well, early bacteriologists classified according to shape, arrangement, growth characteristics, and habitat, and we still do that today. But some of the newer methods include biochemistry, what's happening at the metabolic level in these cells, what is their genetics, what are some of the molecular traits that we find in them, and again, what about their ribosomes? Evolutionary relatedness is most accurately determined through comparison then of the nitrogen bases in the ribosomal RNA. These tend to remain stable in the nucleic acid content over long periods of time during the evolutionary process. So any major differences in the signature sequences indicates distance in ancestry. Now, one of the things that we use as microbiologists is a book called Bergey's Manual of, that should be of, Determinative Bacteriology. This is the definitive published source for bacterial and archaea classification. This looks at phenotypic traits that were based for early classification, such as shape, cultural behavior, and biochemical reactions. It's still used today by clinical microbiologists and researchers who need to quickly identify unknown bacteria. Basically, Berkey's manual has everything that we know as of today about bacteria and some of the archaea. Currently, we're in the 9th edition, but I think the 10th edition is coming out shortly. Another book that we use is Bergey's Manual of Determinative Bacteriology, and this is based entirely upon phenotypic characteristics. This is not as commonly used as the other Berkey's Manual. But this characterizes bacteria by traits commonly assayed in clinical teaching and research labs. It's widely used by microbiologists who need to identify bacteria but don't really need to know about their evolutionary backgrounds. It is used for students of medical microbiology, but most of the time, again, we have this all also included in Berkey's manual, and so we typically tend to use the other book. If you'd like to see a copy, you can come by my office at any time, as I have one of these in my office. And then Berkey's Manual of Systemic Bacteriology is a comprehensive view of bacterial and archaeal relatedness. This actually combines phenotypic information with the ribosomal RNA sequencing for classification of these organisms, and it's actually now available online. Those three: the gram-negative, gram-positive, bacteria without cell walls, and then the archaea. Then we can subgroup them in by the cell shape, by their arrangement, by their oxygen use. Are they aerobic, so they use oxygen in their metabolism? Are they anaerobic, so they don't use oxygen in the metabolism? Or are they facilitative anaerobes, which may or may not use oxygen in their metabolism? And we'll talk more about this when we get to the metabolism chapter. Bacterial species are a collection of bacteria which share an overall pattern of similar traits. So, E. coli. When I say E. coli, well, I'm talking about a species of bacteria which has a huge number, almost I think around 600 different types of E. coli are out there in the environment. So sometimes we talk about subspecies, strains, or types. And these are bacteria of the same species, but have differing characteristics. Serotypes are representatives of a species that stimulate a distinct pattern of antibody response in immune systems by distinct surface molecules. So, for example, E. coli 0157H7 is a strain of E. coli, but it's also a serotype because it has a specific polysaccharide called the 0157 lipopolysaccharide that's found in its cell wall. The H7 actually refers to a specific form of the flagellin protein found in its flagella. So, not only is it a strain, but it's also a serotype. So last concept check. Primarily, bacteria and archaea have been related by comparison of their what? And which Berkey's manual would probably most be often used in a hospital clinical laboratory? And what is measured to determine serotype? And that's it for this chapter. So see you next